Hi, my name is Tatum Liparulo. I am from the Shoemaker Immunosystems Lab in the Department of Chemical and Petroleum Engineering at the University of Pittsburgh. And today I will be presenting my work. Mathematical modeling suggests that monocyte activity may drive sex disparities during influenza infection. Females of reproductive age are at higher risk for severe influenza infection when compared to their male age-matched counterparts. As you can see in this graph, I'm showing a female to male hospitalization ratio. This data comes from the 2009 H1N1 pandemic in Japan. And as you can see, between the ages of roughly 25 to 65, that there is a ratio greater than 1, indicating that females are being hospitalized at greater rates than males, in particular for H1N1 influenza. There are several factors that could explain different infection outcomes between males and females. One of them is sex chromosome-linked genes. For example, there is a protein recognition receptor called TLR7 that helps initiate the innate immune response to pathogens like influenza. It is located on the X chromosome, and since biological females, have 2 copies of the X chromosome, there tends to be a higher expression of this receptor and an increased innate immune response in females compared to males. Additionally, sex hormones have been shown to have a considerable impact on inflammation, and inflammation is a major driver of disease severity in influenza, where it tends to be cases of excessive inflammation that cause severe influenza disease. So influenza infection and the innate immune response interact through complex mechanisms. So simply, the presence of influenza in the lungs initiates an immune response from the cells in the lungs. And what happens is there are a bunch of different mechanisms that are activated that end up resulting in lung inflammation. This immune response in turn helps block the spread of influenza within the lungs and also helps to resolve infection. However, inflammation can very easily become too much, particularly in females compared to males, and this can lead to that increase in hospitalization rates that I've shown before. What's interesting is that there is not an observed difference in influenza viral titers between male and female mice, which further implicates inflammation and immune dysregulation as a cause for disease severity and not just the virus itself. So what I'm showing here is data reconstructed from Robinson et al., published in 2011, of male and female mice infected with H1N1. As you can see in this weight loss chart, the female mice had significantly more severe influenza infection than the male mice, despite being given the same initial viral titer. Additionally, none of the female mice survived past 13 days post-infection, whereas about 50% of the male mice did survive and rebounded to their initial body weight. However, you can see on the right that there is no statistically significant difference in viral titers between 1, 3, 5, and 7 days post-infection for the male and female mice. So what we wanted to do in our work is to utilize mathematical models of ordinary differential equations to see if we can answer some questions about the kinetic differences in the rates controlling sex differences in disease severity, so that we could potentially identify a mechanism that could explain what we see in terms of disease severity that isn't resolved just looking at viral titers. So the first thing we did was identify a suitable publicly available data set in the literature. We identified Robinson et al. 2011 as a suitable data set for what we were setting out to do. The data is sex disaggregated, so male and female data is separate, and there are viral titers and cytokine and chemokine data. Additionally, as I presented on the previous slide, this is that same data. There is a clear male and female sex difference in disease severity. So I'm not going to go over the weight loss and the virus titer data since I showed it on the previous slide, but I will go over these 2 new graphs I'm showing at the bottom. So first, we're using IL-6 fold change data as a marker for interferon, where there is a clear sex difference and an increase in female concentrations of this marker at 5 days post-infection. Additionally, we're using CCL2 MCP1 fold change data as a marker for monocytes at the site of infection. And as you can see here, at 3, 5, and 7 days post-infection, there is a significant increase in this marker in the female mice compared to the male mice. I want to note here that the reason we have to use markers for interferon and monocyte is that the data in this study that we did not complete was limited and incomplete in some ways. So we're really making do the best that we can with data that was just available already for us to use. The model we used was developed by Ackerman et al. 2022, who was a student in our group, and it is a minimal mechanistic model of the innate immune response to influenza infection. So briefly, virus in the system causes interferon to be produced. Interferon in turn causes monocytes to be recruited or produced. The interferon and the monocytes can both block viral growth, and monocytes can also in turn produce interferon. One thing I want to note here is that we are not including any host target cells or target or infected cells in this model, and that is because there is just a lot of uncertainty in terms of the number and proportion of target cells that become infected or resolve infection over this limited 7-day time frame. And we wanted to be very confident in our parameter estimates, so we didn't really want to add more uncertainty to our model. So here, viral growth is modeled using semi-logistic growth. So overall, there are 3 states for virus, interferon, and monocytes, and there are a total of 13 parameters that we did our best to bound within biological evidence. And our overall hypothesis is that we can use this minimal mechanistic model to help us identify distinct sex-specific rates within our ODEs, and the identification of these sex-specific rates will lead us to identify the mechanisms that could potentially be targeted to help reduce disease severity in females. So non-linear mixed effects modeling can help us to enable model selection in absence of validation data. So what am I talking about here? In order for us to identify these sex-specific parameters, we're essentially going to take our single model and with it create a bunch of different models in which some of the parameters are allowed to take on both a male and a female value during model fitting. So first, I'm going to go over a scenario in which all of the parameters are allowed to be different. And in effect, what we end up with is 2 separate models, a fully male model and a fully female model, in which all of the parameters in each of these models can take on a male or a female value. This is our biggest model in terms of the models that we are generating. And it will result in the largest number of parameters. On the flip side, we can create a model, just one model, in which no parameters are different. So in essence, what we do is we take our initial model and we fit it simultaneously to the male and the female data. So this is our smallest model scenario in terms of our overall models that we're generating. And as you can imagine, there might be some difficulty fitting the data that I've shown before, but this is a scenario in which there wouldn't be any sex-specific parameters. And then finally, we fall somewhere in the middle, where there are some parameters in the male and the female model that are allowed to take on these unique values, while there are other parameters shown here in black that actually have to share a value in both the male and the female model throughout the duration of model fitting. And the reason that we are taking this non-mem approach is that this data, we are lacking validation data. So we're not able to actually validate this model after we just fit it to the data. And this enables us to actually pinpoint which parameters must be sex-specific in order to get an appropriate model fit. So what we're going to do is then we're going to use log likelihood to evaluate goodness of fit, but also to calculate the Bayesian information criterion. So briefly, the log likelihood is what we're using as our error function. It's comprised of a number of data points, the error, so the difference between the data and the model, but then also the standard deviation of the data. This is a pretty straightforward equation here. But we're using the log likelihood to feed into a BIC Bayesian information criterion calculation. And we're using the BIC to actually determine which of these models is our best model. And the reason we chose BIC is because there includes a penalty term for a larger number of parameters in the model. And what this does is it penalizes models that have many, many more parameters than another model. And this way we can actually balance goodness of fit with model size. This can help with identifiability issues, but it also gives us increased confidence in the parameter estimates of our overall best model, aka the model with the smallest BIC. So I'm going to go over our sex-specific parameterization. First, we start with that all different condition in which we have a model that is fully male and a model that is fully female fit to the male and female data respectively. As you can see here on the right, looking first at this top row of our male model fit to our male data, that we have a really good fit to our data within about 2 standard deviations. On the bottom here, we have our female model fits to our female data. And we can see here that we also have very good model fit. So allowing all of the parameters to be sex specific and have a male and a female value gives us a good model fit. And we end up with a BIC value of 72. This will be our benchmark value that we'll compare our other model scenarios to. I do want to briefly mention that the y-axes are not the same on the corresponding monocyte, interferon, and viral titer graphs, just when you're looking. Next, we have our none different condition. You can initially see that our BIC value jumps to 321, telling us that this is not an improved model formulation. And we can see here, particularly in the female monocyte fold change best fits, that this is just not an accurate model, indicating that yes, there are parameters within our model that are sex specific. All of these parameters cannot be shared, male and female. Next, we wanted to test the viral production and removal hypothesis, where just the virus itself is sufficient to explain the sex differences we see. As you can see here, again, by a jump in BIC value from 72 to 222, that this is not an appropriate model formulation. And you can see here that really the model is not able to accurately fit the interferon fold change data for the male or the female data. Next, we decided to test monocyte induction and activity as our two sex-specific parameters. We chose these parameters based on the fact that the data of monocyte fold change is so different female to male. As you can see, we actually get an improvement in our BIC value from 72 to 68, and this is telling us that there is strong evidence that monocyte induction and activity is a better model, just those two parameters being sex-specific, than allowing all of the parameters in the model to take on a sex-specific value. And you can see here by looking at the yellow line on these graphs that it very closely follows our model fit to our all different condition, indicating that it is a good fit. Finally, we wanted to see if just monocyte induction as a sex-specific parameter alone was an appropriate model formulation. And as you can see here with a BIC value of 110, it was not. So overall, our parameterization revealed that monocyte induction and monocyte activity are sex-specific parameters within our model. Next, we used Markov chain Monte Carlo, which is an algorithm for exploring high-dimensional parameter spaces to determine the overlap of these sex-specific parameters. So it's not enough to just have a model identify that these are sex-specific. We actually want to confirm that these two parameters do not have male and female overlap in our best fits. Markov, MCMC Markov chain Monte Carlo enables us to explore a much wider range of the parameters, of all of the parameters within our model, so that we can more confidently say that these are indeed sex-specific parameters. So as you can see here, looking at our monocyte induction parameter and our monocyte activity parameter, we do have distinct parameter distributions for both of these parameters. For monocyte induction, shown here in blue and over here in our model in blue, we get a lower overall monocyte induction in the male mice compared to the female mice. However, for monocyte activity, we actually get a higher value in the male mice compared to the female mice. And this is suggesting to us that maybe there are less monocytes being recruited in male mice, but the monocytes there are able to still complete their task of removing virus from the system, whereas for the female in the female mice, there might just be too many monocytes being recruited to the site of infection, causing too high of inflammation and increasing disease severity in influenza. Again, these are just hypotheses. We can't prove that with this model, but this is pointing us in a good direction to test. So what we did then is we used our model and simulated some treatment effects, some model perturbations, in order to determine if perturbing either one of these parameters independently could improve or potentially improve infection outcomes. First, changing monocyte induction in the female model from the female to the male best fit value resulted in an overall increase in virus and interferon concentrations, but a decrease in monocyte concentrations. And overall, this treatment effect is probably not desirable. We wouldn't want to go forth with a treatment pathway where there's higher virus and higher interferon, because that could cause an increase in inflammation in the female mice. However, if we change the monocyte activity from the female to the male value within our female model, we can see that we get an overall decrease in virus, interferon, and monocyte numbers in the female mice. And this is indicating to us that if we were to pursue a potential treatment option, that the activity of the monocytes is what we would want to target in the female mice, not the monocyte induction. Thank you all so much for listening. This work is published in Viruses with the following citation. I would like to thank my lab, Dr. Shoemaker, my advisor, as well as our collaborators at the University of Madison, Wisconsin. Also, I'd like to thank our funding sources at the NIH, NSF, and CTSI, and I would be happy to answer any questions that you have.